Hello students and welcome back to Top Scholars. In today's session, we are going to discuss all the important questions that can be asked from basic biology in your upcoming exam. So stay with me. This is going to be a super helpful session for you all. So let's get started with it. So let's first discuss the important questions that can be asked from the topic cell, the structural and function unit of life. Let's quickly also look at the weightage of this topic. From this topic, children, very short answers kind of questions, that is 2 markers are expected from here. Okay, so let's look at the questions. Let's look at the MCQs. The organelles that contain their own DNA are mitochondria and Golgi bodies. Option B, chloroplast and mitochondria. Option C, ribosomes and vacuoles, and option D, lysosomes and mitochondria. Let's look at the solution. Children, chloroplast and mitochondria, these are the only cell organelles in addition to nucleus who have its own personal DNA. So we can see over here in the chloroplast, we can see this DNA, and also in the mitochondria, we can see this DNA. Okay, so there are total 3 cell organelles that has its DNA. One is the most obvious, that is the nucleus, and along with the nucleus, we also have chloroplast in plant cell and mitochondria in plant as well as animal cell both. However, children, if you look at the structure of DNA, it is slightly different. In nucleus, the DNA is usually linear. However, if you look in the chloroplast and the mitochondria, the DNA are circular, right? So, this is one small difference that we need to keep in mind. Now, children, you might think that why only these 2 cell organelles have DNA of their own and what advantages does it provide? Since chloroplast and mitochondria, they have its own personal DNA, they can replicate on its own without waiting to get any command from the nucleus. Yes, so chloroplast, children, when the conditions are favorable, it will quickly replicate and have more number of copies. From 1, it can even have 10, some may even have 15. And same with the mitochondria. So mitochondria, especially in the muscle cells, they replicate very fast. And we usually see athlete's muscle have more mitochondria in each cell as compared to a normal individual's muscle cell. Alright, and why is that possible? Because inside a cell, this mitochondria can simply replicate on its own without getting any command from the nucleus. Alright, so children, the correct answer over here is option B, that is chloroplast and mitochondria. Let's look at the other cell organelles, that is the Golgi bodies, ribosomes, vacuoles and lysosomes. These cell organelles do not have their own DNA, therefore they cannot replicate on its own. Only when nucleus commands it to replicate, it will be replicate. Okay, now let's move on to the next question. A plant cell is placed in a solution which causes the vacuole to shrink first before the cytoplasm. This solution is most likely going to be hypotonic, option B hypertonic, option C distilled water, and option D isotonic. Okay, children, so now let's look at the solution. So we can see over here 3 options are given where a cell is placed in an isotonic solution. In option B, the cell is placed in a hypertonic solution, and in option C, the cell is placed in a hypotonic solution. Okay. Now, when a cell is placed in a isotonic solution, children, the cell becomes flaccid because there is not much movement of water from osmosis, as the level of water in both the solution and in the cell is almost the same. However, in hypertonic solution, children, because of osmosis, the water will exit the cell and the cell will be plasmolysed. Alright. In hypotonic solution, children, because of osmosis, the water will enter the cell and the cell will become swollen or turgid. Therefore, as per our question, the plant cell which is placed in a solution will cause the vacuole to shrink first before the cytoplasm. This solution will be hypertonic solution. Alright? Alright children, now let's look at this MCQ. During an experiment, a scientist selectively destroys the endoplasmic reticulum, however, still keeps the ribosomes active in it. Because of this, the cell will face difficulty in which of the following processes? Option A, producing amino acids. Option B, folding and transporting the proteins. Option C, breakdown of the waste materials. And option D, expelling of the carbon dioxide. Alright children. So, the endoplasmic reticulum, we know that it's a cell organelle that connects the nuclear membrane with the plasma membrane, right? And on the endoplasmic reticulum, we have ribosomes attached to it, and therefore we call it as the RER, that is the rough endoplasmic reticulum, right? Now, in the question, they have destroyed the endoplasmic reticulum, but they have kept the ribosomes intact. And we have to find out the cell will face difficulty in which process. So, option A says that the cell will not be able to produce amino acids. Alright children, this is absolutely incorrect because the function of the ribosomes is not to produce amino acids. Okay. Option B says folding and transporting of the proteins. Now, this is a correct answer. Let's understand that. So, we need endoplasmic reticulum to modify, to package and transport the proteins. See, protein synthesis takes place inside the ribosomes, alright, but then this protein enters into the endoplasmic reticulum where it will help in packaging, modifying and transporting the proteins outside. So, if we destroy the ER, we will be facing difficulty with the folding and transporting of the proteins. Therefore, option B is correct. Now, let's see why option C and D are incorrect. C says that breaking down of waste material will not be possible. Children, this is not a function of the ER. So, if we destroy the ER, we will not face this problem. Alright. And similarly, expelling carbon dioxide. This is again not the function of ER. Therefore, option C and option D are incorrect. Now, let's look at the next question. A scientist discovers a chemical that can destroy the cristae of mitochondria. Identify the immediate effect. Before we go into the options, let's quickly understand the cristae. Children, cristae are these finger-like foldings which we find in the cavity of the mitochondria. Alright, and in a question, the scientists have destroyed this cristae. So, what may happen? Option A, no DNA replication. Option B, no enzyme secretion. Option C, fall in ATP production. And option D, burst of the lysosomes. Now, children, let's look into the solution. Children, these cristae have this green bead-like structures. Can you see? Alright, these are the DNA strands. And along with that, in the cristae, we have certain particles which are known as the F1 particles. These particles, children, they are involved in the production of ATP. So, if the cristae is damaged, the F1 particles will be damaged, and if F1 particles are damaged, the ATP production will also reduce. And therefore, the correct answer is option C, that is fall in ATP production. Alright children, let's see why the other options are incorrect. Children, the cristae is not responsible for DNA replication. Therefore, any damage to cristae will not affect DNA replication. Again, they are not involved in any enzyme secretion. So, again option B is not correct. And burst of lysosome have nothing to do with mitochondria, as lysosomes are different organelles present in the cell own right. Therefore, the option C is the correct answer. Now let's move to the next question. Assertion: a cell with a defect in Golgi body may affect the lysosome formation. Reason: lysosomes originate only from the Golgi body in the eukaryotic cells. Let's look at the options. Option A, both assertion and reason are true, and reason is the correct explanation of assertion. Option B, both assertion and reasons are true, but reason is not the correct explanation of assertion. Option C, assertion is true, but reason is false. And option D, assertion is false, but reason is true. So let's quickly solve this question. Children, the Golgi complex is a very important part of our cell, right? It gives rise to the primary lysosomes. And this lysosome, children, as you can see, they are filled with this digestive enzymes. That means the lysosomes are birthed from the Golgi apparatus. Therefore, if a cell has defect in the Golgi complexes, the lysosome formation will be affected. Assertion is correct. And the reason for that is the lysosomes originate from the Golgi body, right? So, both the assertion and reason are true, and the reason is the correct explanation of the assertion. So, option A is correct, right? Now, let's move on to the next question. Assertion: the cell wall in a plant cell allows only certain substances to enter and leave the cell without hindrance. Reason: the cell wall is freely permeable. Option A, both assertion and reason are true, and the reason is a correct explanation of assertion. Option B, both assertion and reason are true, but reason is not the correct explanation of assertion. Option C, assertion is true but reason is false, and option D, assertion is false but reason is true. So let's solve it. Children, a cell wall is made up of two membranes, that is the middle lamella and the primary cell wall, and they themselves are composed of pectin, cellulose microfibril and hemicellulose. This structure of the cell wall makes it freely permeable. What does this structure makes it? Freely permeable. That means, let's look at a plant cell. This is the cell wall of the plant cell, and this is the plasma membrane of the plant cell. Alright. So, through this cell wall, everything can enter in the cell, that is water, ions and other substances, and similarly everything can exit out as well. Alright, so cell wall is a freely permeable membrane. However, the cell membrane is semi-permeable. That means it only allows selective substances to enter or exit the cell. Alright, let's look at our question again. The cell wall in a plant cell allows only certain substances. That means the assertion is saying that cell wall is semi-permeable. Right? And the reason over here they have told that the cell wall is freely permeable. So, the correct answer is assertion is false but reason is true. Alright. Who is semi-permeable in the plant cell? It is the cell membrane. Alright, now let's move on to the next question. Alright students, now let's look at this next question. Okay, now let's look at this question. Diagram based fill up. The labeled part A plays a role in dash synthesis by forming and storing dash. Over here we can see that this is a nucleus, and the labeled part is this dense body present inside the nucleus. And what is that? Yes, it is nucleolus. So, in the question, they have asked us that the nucleolus play a role in dash synthesis, and it forms and stores something which helps in the synthesis. So, the correct answer over here is that, children, the nucleolus play a very important role in ribosome synthesis, and for that they form and store the rRNA. What is rRNA, children? rRNA is ribosomal RNA, which along with some proteins forms the ribosomes. Okay, children? So, children, we can see over here nucleus and we can see a enlarged version of the nucleolus. They play important role in formation of ribosomes, which are the protein factories in the cell, and for that they need the raw materials which they form and store, and the raw material is the ribosomal RNA. Alright, children. Now let's look at the next question. In the given diagram, the structure labeled A is dash and it contains the pigment dash. We can see that this is a diagram of chloroplast, which is a plastid. And this disc-like structures, they are the thylakoids, which are green in color because of the pigment chlorophyll. Alright, so the correct answer over here is thylakoids. Now let's look at a next similar question. The given diagram is of the dash and it gives rise to dash in the sperm cell. So this diagram is that of a Golgi complex or a Golgi body, also known as Golgi apparatus, and it gives rise to the acrosome of the sperm cell. So if you look at a sperm, the sperm is wearing a cap at the top. This is the acrosome of the sperm cell and this is the tail. So this acrosome is derived from the Golgi body. Okay. Now let's look at the next question. This diagram shows a large fluid filled organelles which are called as dash and it is surrounded by a membrane known as dash. Children, over here we can see that this is an animal cell, and in the animal cell, fluid filled cell organelles are known as the vacuoles. So these are the images of the vacuoles, and the membrane that it is surrounded by is known as the tonoplast. Alright, so let's look at the solution. As we can see over here, this large fluid filled structures are the vacuoles. Now let's look at this case study, which can be asked for 3 marks. While experimenting, a student cuts an amoeba into 2 unequal parts. He observed that part containing the nucleus survives and grows, and the other part who does not have a nucleus dies. What conclusion can be drawn from the role of the nucleus from this experiment? Question 2. Suggest one vital activity that the nucleus controls in amoeba. And question 3. What might happen if the mitochondria are removed during this activity? So let's solve these questions. First, children, we can see over here, this is the amoeba. Alright, these are the finger or the foot-like processes which are known as the pseudopodia. In here, we can find the food vacuole where digestion will take place. Here we can see this is the main nucleus, that is the main cell organelle of the cell. Alright. This amoeba, children, when we cut this amoeba in an unequal way, that is one part, here we can see this left part, that is the A part, has a nucleus in it and B part does not have a nucleus in it. We find that only the A part survives while the B dies. And the reason for that is the A part has the nucleus. This will help the amoeba regrow in shape and size. However, the B part does not have a nucleus, and as a result of which it cannot survive on its own. The second question that was asked, that what is one important function of the nucleus? So children, nucleus helps the amoeba to control a lot of metabolism in the body. Along with that, nucleus also help in reproduction in amoeba, that is by cell division. Alright. The third question they had asked was, if we remove the mitochondria during this activity, what would happen? Children, if we remove the mitochondria from the amoeba will not be able to produce enough ATPs, and without enough energy, the growth of the amoeba will be slowed down. Alright. Question A, the answer to that was that nucleus was present and therefore that part of the cut amoeba will grow. Question B was What are the function of the nucleus? The nucleus helps in controlling metabolism and it helps in reproduction of amoeba, that is through cell division. And the question 3 was, if we remove or reduce the number of mitochondria, then what will happen? Then the amount of ATP production will also reduce. There will be low energy and there will be therefore slow growth. Alright children, now let's move on to the next question. The diagram below shows an animal cell. Identify the structure which is labeled as A. So we can see over here, these are the structures that is giving rise to the spindle fibers, right? We can see the spindle fibers moving from top of the cell to the bottom of the cell. These structures are centrosomes. Question B. State its important function. It helps in development of the spindle fibers, that helps in positioning the chromosomes in the equatorial plate or in the midline of the cell. So they help in rearranging the chromosome on the equatorial plate. That is its important function. So let's quickly write it down. It helps in rearranging and positioning at equatorial plate during metaphase. So let's quickly look at the solution, children. The label structure is centrosome and it plays an important role in spindle formation. Okay. Now let's move to the next question. In a diagram, the nucleus is centrally located, but a small vacuole is present at one side. Is it a plant cell or an animal cell? Question B. Which labeled part supports your conclusion? So let's look at the solution. Children, it's an animal cell, and the reason that I can so strongly tell this is because of the presence of centrosomes. These perpendicular bar-like structures, they are only and only present in the animal cell and completely absent in the plant cell. And that helps us bring me to the right answer. Alright. Now let's look at the next question. A student places amoeba cells and onion cells in a solution that has less solute than the cell sac. After some time, the onion cell remains safe, but the amoeba cells, they are at a risk of bursting. Explain why. Let's look at the solution, children. Children, over here, we have placed the amoeba and the onion cell in a hypotonic solution. So over here, this is the amoeba in a hypotonic solution and this is the onion cell in a hypotonic solution. Now children, what happens is that because of osmosis, the water will move from the solution into the amoeba, and as a result of which the amoeba will start swelling in size. And same thing will happen over here, that the water will start moving inside the onion cell. However, the swelling of the onion cell will be limited. It will not be at a risk because we have a very strong cell wall. You can see over here, children, the onion cell, it has a very thick, very protective and a rigid cell wall. It will not allow the onion cell to burst open so quickly because it will limit and it will take care of that pressure. However, in amoeba, since it's an animal cell, it does not have a cell wall. That means there is no rigidity, there is no protection. The more the water enters inside the amoeba cell, the more it will be at a risk after a time and it may completely burst. Alright, so the difference is of the advantage and the presence of a cell wall that amoeba does not have, but a plant cell has. Alright, now let's move to the next question. Look at this given image below. Identify the structures which are labeled as A and B and evaluate how the absence of each structure affects the functioning of the cell. Let's look at the solution. Children, A over here is standing for rough endoplasmic reticulum. We can clearly see that endoplasmic reticulum is a cell organelle that connects the nuclear membrane to the cell membrane. So here we can see this is the nucleus and this is its membrane. So the nuclear membrane is giving rise to the endoplasmic reticulum. Okay. Over here also there is endoplasmic reticulum. But if you see over here, on this endoplasmic reticulum, we see there are lots of ribosomes attached to it, and therefore this is the rough endoplasmic reticulum. Whereas the still connected with the nucleus, it does not have any ribosomes attached to it, and therefore label B is the smooth endoplasmic reticulum or SER. Children, RER is required for protein synthesis, whereas SER is required for lipid synthesis. So, absence of any of this cell organelle will reduce the synthesis of that particular chemical. So, if RER is absent, the protein synthesis in a cell will go down, and if SER is absent, then the lipid synthesis in that cell organelle will go down. Alright. So, now let's go to the next question. Which plastid contains chlorophyll for photosynthesis? Name a plastid that stores starch. So, let's look at the solution. Children, we can see over here two plastid, that is chloroplast and the leucoplast. Chloroplast is a green color plastid, that means it is involved in photosynthesis. We will find it in the stems and the leaf cells, and that will perform photosynthesis and produce food. Whereas leucoplast, this is a plastid which we will find in the potatoes especially, because it stores a lot of starch. So, potatoes are filled with leucoplast. Okay. So, this is a starch storing plastid. Now, let's go ahead. What type of movement do white blood cell shows? Name the cell that controls the opening and closing of stomata. Also state their shape and one more function. Children, WBCs or the white blood cells, they are also known as leukocytes. They are the only cells who can actually exit a capillary and move into the tissue spaces. Yes, RBCs and platelets, they are not able to do so, but only WBCs can do so from small spaces present between the cells of capillaries. So, let's look at a capillary quickly, children. This is a small capillary, alright. This is one cell of a capillary, this is a second cell of a capillary. This space between the two cells of the capillary is known as the endothelial pore. And it is through this space that the WBCs, just like a amoeba, will squeeze out of the capillary, and this process through which it squeezes out is known as diapetesis, which is similar to a amoeboid movement. Okay. Now coming to the second question, that is about the cells that guards the opening and closing of the stomata. Children's stomata, as we can see over here, this is the opening which we find in the leaves, right? And it is guarded by these cells which are present on this side in pairs. The cells that guard the opening of stomata are also known as the guard cells. What shape do they have? Children, it's very clear. They are kidney shaped. What roles do they play? They play a role in opening and closing of stomata. So, when the stomata closes, the gaseous exchange will stop, and when it opens, the gaseous exchange will continue, right? So, over here, gaseous exchange will take place. Over here, it is closed. We cannot see any opening. We cannot see any stomata. So, here the gaseous exchange will stop. What another function that they play a role? Obviously, first is a gaseous exchange. They remove the excretory products like CO2 and O2. CO2 is a gaseous waste produced during respiration, and O2 is a gaseous waste which is produced during photosynthesis. That will be removed through stomata. Other than that, children, even through stomata, there is water loss, that is transpiration, right? And that will help in the movement of water from the root towards the stem. So, this is the second function of the stomata. The first function, as we discussed, is the gaseous exchange, and the second function, children, is it helps in the transpiration pull. That is a movement of water from the root towards the stem and leaves due to constant loss of water from the stomata. Alright, children, I hope this was clear. Now, let's look at the next question. During a biological practical, Prachi observed a cell from an onion peel and a human cheek. She tried to compare both the cells and note down the observations. Questions. A. Name one plant feature which is absent in animal cell. Question B. Name one animal cell feature which is absent in plant cell. Question C. Name one similarity between the plant cell and the animal cell. So let's solve it. So one plant cell feature which is absent in the animal cell is the cell wall. This is a very important characteristic of a plant cell, that it has an outer rigid, protective and supportive cell wall that animal cells don't have. One animal cell feature which is absent in plant cell is centrosome. And one similarity between plant cell and animal cell will be presence of the cell membrane. Alright children, now let's move on to the next question. Observe the diagram of a plant cell below and match the label parts 1 to 4 with their correct functions. Alright, so the function we are given is this cell organelle, it controls the activity of the cell and contains the genetic material. Children, the correct answer will be nucleus. Question B. Provides rigidity and structural support to the plant cell. The correct answer will be the cell wall. Question C. Controls the entry and exit of substances. The correct answer is the cell membrane. Question D. It conducts photosynthesis to make food. The correct answer is chloroplast. And the last question, it stores water, nutrients and waste. The correct answer is the vacuole. Alright. Okay children, now let's discuss the important questions that can be asked from this topic, that is the structure of chromosomes, cell cycle and cell division. First, let's look at this MCQ. Which phase of the cell cycle is sometimes called as the resting phase? Pay attention to this word. Option A, synthesis or the S phase. Option B, interphase. Option C, G1 phase. And option D, mitosis phase. Okay children, let's look at the solution. Children, when we look at a cell cycle is divided mainly into two events. Let's quickly note it down. That is the interphase and the mitotic or the dividing phase. The mitotic or the dividing phase, children, this is the phase where the cell actively divides. That means obviously this cannot be the right answer. Alright. Interphase is also known as the resting phase. Although this is a misnomer, children, please keep in mind, interphase was earlier known as a resting phase, but now we do not consider this as the resting phase because the cell is highly active during this phase. See children, after interphase, the cell undergoes into the mitotic or the dividing phase. That means the cell will actively divide to give rise to two daughter cells. That means in the interphase, the cell actively needs to produce all the raw materials and produce new copy of DNA and other cell organelles, so that when the two daughter cells are formed, both the daughter cells have equal number of DNA and the cell organelles. And therefore, the cell is highly active in the interphase, which was previously known as the resting phase. If we can see in this diagram over here, children, the interphase consists of further three more phases. That is the G1 phase, the S phase and the G2 phase. So, everything from here, children, you can see from here, this entire is the interphase. Okay, so here we can see in the interphase, there is a G1 phase, there is a S phase and there is a G2 phase. Alright, so the correct answer is the option B, that is interphase. Alright students, now let's move on to the next question. The sister chromatids are visible for the first time in which phase when the cell is undergoing mitosis? Let's read the question one more time. It's an important and slightly tricky question. The sister chromatids are visible for the first time in which phase when the cell is undergoing mitosis? Let's read the question one more time. It's an important and slightly tricky question. The sister chromatids are visible for the first time in which phase when the cell is undergoing mitosis? Let's read the question one more time. It's an important and slightly tricky question. They are visible for the first time in which phase? Alright, let's look at the option. Option A is prophase, option B is metaphase, option C is anaphase, and option D is telophase. Okay, now let's look at the solution. Children, we know that when a cell undergoes cell division, the first subphase of the mitotic phase is the prophase. Now, before the prophase starts, we know that inside a cell and inside the nucleus, the DNA is present in a form of a thread which is known as a chromatin network. And when it undergoes into prophase, the condensation of the DNA happens. Condensation of the DNA means that this long thread of DNA, it starts shortening in length and therefore it becomes short and also thick. Alright. So, by the end of prophase, where the condensation ends, this DNA appears like chromosomes. And not only that, children, remember in the S phase we had formed an extra copy of the DNA. Now, that extra copy of the DNA will also be visible in the form of the sister chromatids. Alright. Therefore, children, the correct answer over here is that the sister chromatids will be visible for the first time in the prophase. That is option A. Alright, children. Now, let's move on to the next question. A scientist, while experimenting, removes 2 chromosomes from a plant cell that has a total of 12 chromosomes. The cell is then allowed to undergo meiosis. At the end, each daughter cell will have how many number of chromosomes? Let's look at the option. Option A is 10, option B is 12, option C is 5 and option D is 6. Now let's look at the solution. Children, we know that meiosis is a reductional division, right? What do you mean by reductional division? The number of chromosomes in the gametes which are formed, it will be half as compared to the number of chromosomes in the parent cell. Now, the data given to over here is that total the parent cell had 12 chromosomes, but scientists removed 2 and therefore now the parent cells only have 10 chromosomes. Now, this parent cell with 10 chromosomes undergoes meiosis. So, let's draw the diagram over here. This is a parent cell having 10 chromosomes. Now, it undergoes meiosis 1. We know that meiosis 1 is a reductional division. That means the daughter cell that will be formed from this process will only have 5 chromosomes, that is half of the parent chromosomes. Now, after this meiosis 1 is completed, each daughter cell will undergo its own meiosis 2. Now, meiosis 2 is an equational division, meaning the daughter cell that will be formed from this meiosis will have the same number of the parent chromosomes. Therefore, children, I hope you got the answer. So, the correct answer will be option C, that is 5. That when only 10 chromosomes were left in a parent plant cell, after meiosis, in the gametes, we will only find half of its number, that is 10 divided by 2 and that will be 5. Alright children, I hope this was clear. Now, let's move on to the next question. Which events are represented in the given image? Let's look at the option. Option A is anaphase and metaphase. Option B is telophase and cytokinesis. Option C is prophase and metaphase. And option D is prophase and metaphase again. The correct answer over here is, children, are you able to guess it? If not, let's look at the solution. Children, in this diagram, we can clearly see 3 major changes. And these 3 major changes are, first, appearance of the nuclear membrane in both the daughter cell. Here we can see even in this daughter cell, the nuclear envelope or the nuclear membrane is reappearing. So, this is the first change. The second chain, children, that we can see is this structure, that is the nucleolus. So the nucleolus has also reappeared. Children, these two changes are usually seen in the last phase of division, that is the telophase. Remember, telophase is reverse prophase. So whatever changes that happened in the prophase, now those changes will return back. In the prophase, children, I hope you remember that the DNA condenses to form chromosomes, the nuclear membrane starts to disappear, and the nucleolus starts to disappear. And all of these three things are returning over here. This means this is a telophase. Now, children, the cell divides so fast that telophase usually happens simultaneously with cytokinesis. So, we can see over here that there is this cleavage or the furrow formation. What we can see over here? We can see over here this cleavage or the furrow formation, where the cell membranes are pinching towards the center and thus separating two daughter cells. That means along with the telophase, we are also seeing cytokinesis. Also, this image gives us one more information, is that this is a dividing animal cell and not a plant cell, because only animal cell show cleavage furrow method of cytokinesis. Alright, therefore the correct answer over here will be option B, that is telophase and cytokinesis. Right? Alright, children. Now let's move to the next question. Assertion: DNA replication is called as a semi-conservative method. Reason: each new DNA molecule contains one old strand from the parent and another strand which is newly synthesized. Let's look at the options. Option A, both assertion and reason are true and reason is the correct explanation of assertion. Option B, both assertion and reason are true but reason is not the correct explanation of assertion. Option C, assertion is true but reason is false. And option D, assertion is false but reason is true. Let's look at the solution, children. Children, DNA replication, it happens when a cell decides to divide and form daughter cells, right? And when DNA replication happens, children, the DNA strands, they separate from each other. So let me show it to you. If this is my parental DNA, all right, during replication, these strands will separate from each other and these strands will be used as a template strand on which this new strand of DNA will be synthesized. And eventually, children, the two DNA strands that will be formed will have one as the parental strand and the other which is the newly synthesized strand. This DNA will go to daughter number 1. Alright, this will go to the daughter cell number 1 and this DNA will go to the daughter cell number 2. So what we can see over here, that each daughter is receiving one strand of the parent itself. Look at it. So it is receiving one strand of DNA from the parent as it is, and the other strand is newly synthesized. And such a kind of DNA replication is known as semi-conservative DNA replication. Alright, so in the diagram also, children, we can see over here the blue strand is the parental strand and the red strand is a newly synthesized DNA strand. Alright, so we can see over here, now when you look at this first generation, you can see the red and blue strands both. That means there is one parental strand and one newly synthesized strand in all the daughter DNA. Alright, this method is known as semi-replication method. So now let's come back to our question. So the assertion is that DNA replication is called as semi-conservative, which is correct. And each new DNA molecule contains one old strand and one strand which is newly synthesized. This is also correct and it is the correct reason for the assertion as well. Therefore, the correct answer is option A, where both assertion and reasons are true and reason is the correct explanation of assertion. Alright children, quite simple, right? Good. Now let's move on to the next question. Question number 6, again it's an assertion reason kind of a question. Let's read it. Assertion is, animal cell uses a cleavage furrow method during cytokinesis. Reason, animal cells have cellulose wall that can be divided by a furrow. Let's look at the options. Option A says both assertion and reason are true and reason is the correct explanation of assertion. Option B says both assertion and reason are true but reason is not the correct explanation of assertion. Option 3 says assertion is true but reason is false, and option D says assertion is false but reason is true. So let's look at the solution. Children, first of all, animal cells, they are different from the plant cell, and the one major difference is the presence of the cellulosic cell wall. What is the one major difference? The presence of cellulosic cell wall. Children, this cellulosic cell wall, that means a cell wall which is made up of cellulose. We can see over here this dark green structure. This is a very rigid structure and therefore it cannot pinch inwards or it cannot bend or fold inwards. And therefore, the plant cells, they cannot divide like an animal cell do. That is by the membrane going inwards, pinching and separating the two daughter cells. The plant cell needs to have a different method, that is a cell plate formation method. So you can see over here there is a cell plate which extends from the center towards the periphery. It extends from the center towards the periphery and thus separating two daughter cells. Whereas in animal cells there is no cell wall and the cell membrane is very flexible, and as a result of which the cell membrane can move inwards, it can form that furrow and separate the two daughter cells. Alright, so please remember the animal cell, it divides by cleavage furrow method, whereas the plant cells divides by cell plate method, and the reason for that is rigid cell wall. Alright, I hope this is clear. So let's look at the option. So we know that assertion A is correct, but the statement of the reason that the animal cells have a cellulose wall, that is incorrect. Therefore, the correct answer is assertion is true, but the reason is false. Easy, children? Yes? Alright, let's move on to the next question. Now we are going to fill up in the diagrams. So let's look at this diagram. In this diagram, two identical thread-like structures are labeled A and they are joined together at this blue color point. What is it known as? Alright children. Sabse pehle, children, I hope this is clear that these are the sister chromatids. What are sister chromatids, children? Let me break it down and make it easier for you. Let's draw a simple chromosome. Alright children, now we know that when a cell divides during the S phase, a new copy of DNA is formed. That means a new copy of chromosome is also available and it will be attached to this same point as the previous chromosomes where they were attached. Children, now these are referred as the arms of chromosomes. These are referred as the arms of chromosomes, which are also known as chromatids. Okay. Now children, these two arms of chromosomes, meaning this chromatid and this chromatid, they carry identical information because it is essentially a copy or a Xerox, right? Therefore, this is sister chromatids. Similarly, children, if you look at the lower arms of the chromosome, these are the lower arms that I am talking about. Whatever information that was present in this original DNA got copied into this DNA as well. Yes or no? Meaning, these two are also sister chromatids. Right? Okay, children? Now, the point where these sister chromatids are connected to each other, this constriction you can see, it is known as the centromere. What is it known as? It is known as the centromere. Okay, children? I hope this is clear. Please do not confuse centromere with centriole and centrosomes. Lot of students confuse between these three words. Centromeres is the primary constriction of chromosome, where the sister chromatids are attached. Centrioles are cylindrical cell organelles present inside a cell that helps in separation of the chromosome during cell division. Whereas centrosomes, it is a cell organelle which is formed when two centrioles, they lie perpendicular to each other, and it gives rise to the astral rays which will form the spindle fibers that will also help during cell division by separating the chromosomes. Okay, so centriole and centrosomes, they are the cell organelles, whereas centromere is a constriction of chromosome where the sister chromatid meets each other. Okay, children, I hope this clears your confusion and also helps you remember the terms correctly. Alright? Alright, children, now let's look at this question. The stage of mitosis which is depicted in the diagram is. So, let's look at the diagram clearly. The first observation that you can make is, first, the centrosomes are at poles. The second observation that you can make over here is presence of spindle fibers. The third observation that we can make over here is that we are seeing chromosomes with its sister chromatid. And the fourth thing that we can observe over here is disappearing nuclear membrane. Children, all these four are characteristics of late prophase. Alright, therefore the correct answer is late prophase. Alright, so what we can see this aster, which is the astral rays coming out of the centrosomes, giving rise to this spindle fibers, and the nuclear membrane disappearing. These are the three main changes that we can see. Now, lots of students may confuse this with metaphase. Why is metaphase not the right answer over here? For two main reasons. In metaphase, the nuclear membrane completely disappears. So, you will not be able to see any nuclear membrane at all. Okay, so your cell would look something like this. This is just the cell membrane that we can see no nuclear membrane. We will see the centriole as it is, we will see the astral rays, yes, we will see all of it. We will also see continuous spindle fibers. We will also see some discontinuous spindle fibers, but which will be attached to the chromosomes. And eventually, as we move ahead in the metaphase, children, these chromosomes, they will come into the equator plate, meaning It will come over here. This is a proper metaphase. Since we do not see the chromosomes at the equator, and since we see still some nuclear membrane, bits of it left behind, this is not a metaphase, but this is a previous phase of metaphase, that is the prophase. And specifically, this is the end of the prophase, therefore it is the late prophase. Okay, students? Now let's move ahead. Now let's look at this question. The given structure is a dash and it is made up of which three parts? Let's dig into this structure. Children, this structure is a monomer of DNA, which is known as the nucleotide or deoxyribonucleotide. Right? Now it consists of three parts. You can see over here a P structure. This is the phosphate group. Phosphate group, children, is attached to the fifth carbon of the sugar. Yes. So this is the fifth carbon of the sugar. That means this is the fourth, this is the third, this is the second, and this is the first carbon. A sugar who has 5 carbons, they are of two types. They are either deoxyribose or ribose sugar. Since we have said that this is a deoxyribonucleotide, that is a monomer of DNA, this sugar will be deoxyribose sugar. Now you might wonder that, ma'am, what is over here at the ground when all the 5 carbons are present over here? Yes, over here we have the oxygen. So that is why this has only 1, 2, 3, 4 and 5 carbon, which is a deoxyribose sugar. On the first carbon, this blue color structure that we can see, this is a nitrogenous base. That is a nitrogen containing compound. Alright, so let's look at the solution. Children, this is a structure of a nucleotide. We have a phosphate group, we have a sugar group, and we have a nitrogenous base over here. Alright, what is a nucleotide, children? Nucleotides, they are the monomers of nucleic acid. Now you might ask, ma'am, what is a nucleic acid? But a nucleic acid can be DNA or a nucleic acid can be RNA. Yes, so just like proteins are big polymers, they are made up of monomers, which are amino acid. Similarly, DNA is also a big polymer and it is made up of multiple monomers, which are known as nucleotides. And when you look at the nucleotide, it itself is made up of 3 structures. So this entire unit, children, this is one single monomer. It is a complex monomer, I agree, but it is one single monomer. Like that, children, in our DNA, we have more than 1 million nucleotides. 1 million is 10 lakhs. So we have more than 10 lakhs such nucleotides coming together and forming our DNA. And trillion cells of our body have DNA of its own. Alright, children. So this is the nucleotide, which is a monomer of nucleic acid. If we consider DNA, that is a deoxyribonucleic acid, then its monomer will be known as deoxyribonucleotide. Alright, and if we consider RNA, then its monomer will be known as ribonucleotide. Are we clear? So DNA is formed by deoxyribonucleotide and RNA is formed by ribonucleotide. So only difference is the prefix deoxy. Otherwise, it's the same word. But it changes a lot of things for DNA and RNA. Just by absence of one oxygen, RNA is changed into DNA. And you know how important DNA is in our body. Alright, without DNA, we are nothing, literally. Anyways, let's move ahead now. Alright, children. Now let's look at the next question. The cells in the given structure undergoes dash to produce pollen. So let's look into it. Here we can see, children, these are the pollen grains. And we know that the pollen grains are produced inside the anther. And this anther is supported by this stalk-like structure which is known as the filament. Anther and the filament forms the stamen. Now you all must be knowing that stamen is the male reproductive part of the flower, right? So let me write it down. Stamen is the male reproductive part of the flower. Right, children? Right? So since the male reproductive part of the flower is stamen, the cells of the stamen will produce the gametes. And we know that gametes are produced from meiosis. And therefore, the correct answer over here is meiosis. Alright, children. So let's look into the solution. Over here, meiosis are of two phases, children. We have the meiosis 1 and we have the meiosis 2. Alright. In the meiosis 1, we can see the prophase 1, metaphase 1, anaphase 1 and the telophase 1, which gives rise to 2 daughter cells. Now, one very important thing that children we need to remember is that the number of chromosome after meiosis 1 becomes half. Therefore, we can see over here that when the cell was undergoing prophase 1, it was diploid. It has 2 set of chromosomes. And as it finished the meiosis 1, the 2 daughter cell that we got, they both have single set of chromosomes and the number have reduced. Now, both the daughter cells will further undergo meiosis 2. So, from prophase 2, it will enter into metaphase 2, into anaphase 2 and eventually into telophase 2. And this will result in the ending of the meiosis 2. And at the end of the meiosis 2, children, we will finally have the gametes which will be present in the pollen grains. Alright. Since gametes are produced by meiosis, the correct answer will be meiosis. Alright, children. Now, let's go to the next question. Let's look at this case study. Scenario. A student observes a root tip cell under a microscope. Some cells show chromosomes aligned at the equator and others shows chromatid moving apart from each other. He counts a total of 43 chromosomes. Identify the phases observed in the onion cell. How many chromosomes and DNA molecules will be present just before the metaphase? Which phase precedes the cytokinesis in the cell? Now, let's look at the solution. Children, we know that the onion cell is not producing any gamete right now. That means it is undergoing mitosis. Alright. Now, we know that in the metaphase, children, we can see that the chromosomes will be aligned with each other. Right. So, the phase where the chromosomes are aligned at the equator is the metaphase. During anaphase, spindle fibers shorten and pull the separated chromatids towards opposite poles of the cell. So, let's look at this diagram, children, that I am going to draw. We know that the spindle fiber, they are attached to the centromere of the chromosomes. So, one spindle fiber is pulling the chromosome down, one spindle fiber is pulling the chromosome up, and they are pulling at the same time. So, that pressure will come on this centromere, because of which the centromere will break and it will take one part of the chromatid towards the upper pole in this direction and one second part of the chromatids towards the lower pole of the cell. So, we can see the chromatids are separated from each other and that phase will be the anaphase. Alright, children. So, let's look at the question. So, the phases which were observed in the onion cell, the first we observed the metaphase, and after that we observed the anaphase. Now, how many chromosomes and DNA molecule will be present just before the metaphase? Alright, let's read the question one more time. A student observes the root tip cells under a microscope. Some cells show chromosomes aligned at the equator and some other shows chromatid moving apart. And he counts total 43 chromosomes in total. So how many chromosomes or DNA molecules will be present just before the metaphase? It will be 43. Alright children? Chalo, now let's move to the next question. Which phase precedes cytokinesis in the cell? Now we know that the cell cycle is divided into two phases. We have the interphase and we have the mitotic phase. Mitotic phase is further classified into two more phases, that is the karyokinesis, that is the nuclear division, and the cytokinesis. The karyokinesis is further classified into four phase children, which is the prophase, metaphase, anaphase, and telophase. So, which phase precedes or which comes before cytokinesis is the telophase. So, the correct answer over here is telophase. Alright children, now let's move on to the next question. Based on the given image, answer the following question. So children, over here we can see the entire structure of DNA. This is one strand of DNA and this is the second strand of DNA. Okay, in the blue we can see that this is adenine, in the green we can see it's thymine, in the orange we can see it's guanine, and in the yellow colored structure we can see the cytosine. Alright, the pink color structures, they represent the deoxyribose sugar, and this green color structure, they represents the phosphate. Alright children, so now let's look at the question. Name the nitrogenous bases which is found in DNA. Next, which two nitrogenous bases are paired with two hydrogen bonds? Very easy question. Let's look at the solution. Children, let's look at this diagram again. So, which are the nitrogenous bases that we find in the DNA? It's given right over here on the left. We find the adenine, thymine, guanine, and cytosine. These are the four nitrogenous bases that we find in the DNA. Alright. In simple words, you can remember A, G, T, and C. Alright. These are the nitrogenous bases of DNA. The second question that was asked, that which two nitrogenous bases are connected or bonded with each other with just two hydrogen bonds. Over here children, let's look at the structure of DNA, right? Where are we finding two hydrogen bonds? We are finding it over here, right? Which are these two nitrogenous bases? Adenine and thymine. So the correct answer to the question number 2 is adenine and thymine. So children, adenine and thymine, they are bonded with the help of two hydrogen bonds. Whereas if you look for guanine and cytosine, we have 3 hydrogen bonds. Okay children, very simple. Now let's move on to the next question. Look at this figure below and answer the following questions. The point where crossing over occurs during meiosis are called. During which division of meiosis does crossing over occurs? So let's answer it. Children, this point where the crossing over of the two DNA is happening is known as chiasma. And during which division of meiosis does it occur? It occurs during meiosis 1. Especially during the Paquetin stage. So let's look at the answer children. So the crossing over is known as the chiasma. This point is known as the chiasma. Alright. I hope this is clear. Children, please remember that crossing over, this phenomenon is only and only seen in meiosis. It helps in exchanging the genetic material between the parental DNA and forming new recombinants new variations in an organism. Alright children, now let's move ahead. A student observes a dividing cell under an electron microscope and observes the thread-like structures. On further analysis, she notes that DNA is wrapped around small protein complexes and it forms a bead-like arrangement. Name these thread-like structures. What are the protein complexes around which DNA is wrapped, and what is this bead-like unit formed by DNA and these proteins called as? So let's answer it one by one. Children, these thread-like structures are known as the chromosomes, which are formed by condensation or dehydration of DNA. Simply when the water is removed, the DNA starts to shorten slightly and they appear thread-like, which are known as the chromosomes. Now coming to the next question. What are the protein complexes around which DNA is wrapped? Children, these proteins are known as histone proteins. Let me tell you a little bit more about the histone proteins and why is it required. Children, our DNA is 2 meters long, okay, and let me give you some visual representation of it. Starting from this one shoulder till the tip of my hand, this distance is roughly 1 meter, okay, this is 1 meter. Now DNA is total 2 meters long, so it is twice this length, right children? Now when the DNA is so long, how will you fit it inside a microscopic cell? It is literally impossible, right? And therefore we need histone proteins. Now let me explain how do they help wrapping the DNA so that the DNA can be condensed and kept inside the cell. So histone proteins, children, they are like ball-like structures, okay? They are simple ball-like structures. Let's imagine a ball and we take a thread, okay? Now on this ball, I'm going to wrap the thread. I'll take this one turn and then I will take another 3/4 turn, alright? And then I will not complete the second turn, I'll just leave it at 3/4 and I will extend this DNA, take another histone protein and do 1 and a 3/4 turn. Again, I'll not complete the second turn, I'll take this DNA ahead and wrap it on another histone proteins and do 1 and a 3/4 turn. So in this sense, don't you think children, I am reducing the length of the DNA? And that's exactly why we need the histone proteins. By wrapping the DNA on the histone proteins, we will reduce its length and we will make it so condensed that we can keep it inside the nucleus. Okay, so the histone proteins are the proteins, these are these balls on which the DNA is wrapped. Alright, these proteins, children, please remember, they are basic and positively charged proteins. And therefore, they attract acidic and negatively charged DNA. Like I told you, opposites attract. So it keeps the DNA wounded on it. Alright. Now this bead-like unit which is formed by DNA and these proteins is. So let me show it to you. Like I told you children, this is a ball-like structure on which I have wrapped DNA to 1 and a 3/4 and then I took a second ball and I wrapped the DNA to 1 and a 3/4 and then I took the third ball and I wrapped the DNA to 1 and a 3/4. So this bead-like structure that I am getting to see is known as nucleosome. What is it known as? Nucleosome. It is a packaging unit of DNA. What is it? It's a packaging unit of DNA. Let's look at its diagram. So children, we can see over here, this is my normal regular DNA and these are these histone proteins. Like I told you, they are like a ball. You can see how this DNA is being wrapped around this histone proteins, which leads to its condensation further and from a DNA we will get a chromosome. Alright, because we have condensed it so much. Over here children, you can see this particular part, how we can see this ball and DNA on the side and then another ball and DNA on the side. So this is a beaded appearance and this packaging unit is known as the nucleosome. Are we clear children? Alright, now let's move on to the next question. Based on the image given below, answer the following questions. Identify the phase where DNA replicates. Which phase prepares the cell for mitosis? Let's look at the solution, children. Over here, we can see this huge diagram of a cell cycle. Alright, children. We can see that this pink color phase, this entire pink color phase, children, this is the dividing or the mitotic phase of the cell, right? And the remaining, this entire phase, children, this remaining phase is referred as the interphase. Interphase is also known as the resting phase. It's a phase, children, where the cell, it accumulates all the raw materials, it produces more proteins, more energy and duplicates the cell organelles as well as it duplicates the DNA as well. Alright, so this is the interphase. Now specifically, in which phase does the DNA replicates? So let's look into it. Children, interphase or the resting phase is further classified into 3 more phases. That is the G1 phase, synthesis phase, that is the S phase, and the G2 phase. Alright children, so in the S phase or the synthesis phase, DNA replication will be seen. So we can see over here, children, this is the phase where the DNA will be replicated. That means I will make another Xerox of my DNA. Why do I need to do it? Because children, we are going to form 2 daughter cells from mitosis and each daughter cell, for it to survive, needs its own DNA. So we need 2 copies of DNA that can be given to both the daughter cells, and its replication or its formation or its synthesis will be seen in the S phase of the interphase. Alright, so this answers your question 1. The second question was, which is the phase where the cell gets ready for mitosis? We have also answered that question. Can you think of it? Yes, you are correct. It is interphase where the cell is getting ready. Alright, let's move on to the next question now. What is a gene? Give 1 significance of mitosis in a multicellular organisms. Let's look at the solution, children. A gene is a specific segment of DNA. So we can see where this is a whole blue DNA which is running, right? This DNA has lots of information which will give rise to a specific characteristic of your body. In simple words, your DNA has information about how your face will be. It has information about how your eyes will look like, what shape will it be, what color will it have, how tall you will grow, how much bigger your limbs will grow. Getting it? So your DNA has information to develop everything of your body. Every part of your body, children, is developed because of the information present in your DNA. Without DNA, you will never develop. You will never look what you look like right now. Yes. Now, this DNA has all this information, but it is not randomly located on the DNA. Every DNA has a specific segment which has a specific information to give rise to a specific part of the body. Like for example, let's say that this part of DNA, it has a information to develop your eye color. So, if you have brown color eyes like me, it is because we both have a DNA that encodes for a brown colored eye. Similarly, let's say this is that segment of DNA that has information to develop hair. So that will give rise to my hair. It will also decide what color it will be. It will also decide whether it will be straight or it will be wavy or extremely curly. So genes, they are this specific segment of DNA that codes for a specific characteristic in your body. However, this is not as simple as we think. It does not directly give rise to the color of my eye. For that, there is some process. Meaning, this segment of DNA or the gene, it needs to give rise to a specific protein by a process which is known as transcription and translation. These two processes will help in formation of a protein, and this protein will help in development of that particular characteristic. Okay, children. So I hope gene is clear. So what is a gene? Gene is a segment of DNA that has information to develop a particular characteristic of your body by producing a protein through process of transcription and translation. I hope this is clear, children. Okay. Now let's come to the second question, which was, what is the significance of mitosis in our body? We know that mitosis do not produce gametes. Gametes are only produced by meiosis, right? Then why is mitosis important? Ever have you experienced cuts on your skin? Like you must be running and you fell and you bruised your knees or you bruised your elbow or your arms, right? And you bleeded for a while. And after some time, a new layer of skin comes over it and completely forms a new skin on it. Yes or no? How did that happen? How did a part where your skin was completely scraped off, you were bleeding, now is suddenly covered with a new layer of cells and skin? That happened because of mitosis. So let me show you this with an example. Let's say this was your skin. Now you fell, and because of which there was this cut on the skin, and because of it there was bleeding. And after some time, children, you will see that this entire cut is completely covered and a new skin is formed on top of it. That is because, children, the healthy skin cells which were next to this injured part, they started dividing. They started doing mitosis, and it formed new cells that completely covered the cut part. So mitosis, it helps in healing of wounds. Mitosis also helps in formation of new cells every day. Let me give you an example. Children, the red blood cells of our body, they only survive for 120 days, that is 4 months. That means at every 4 months, new RBCs have to be freshly produced by the bones. Now, how is that possible? A new cell can only come from an existing cell by cell division. That means there are certain cells in the bone who are dividing at a crazy, insane speed to produce more RBCs. So it also helps in formation of new cells continuously. Alright, so all the cells of our body, not just RBC, children, all the cells of our body, they constantly die and they are replaced with new cells by mitosis. Alright, children? Okay, so in this question we covered what is a gene and we covered the significance or importance of mitosis. Okay, now let's move on to the next question. List the main phases of mitosis and what would happen if the chromosomes do not align properly at the equator during metaphase. Alright, children, let's solve the first question. We know that mitosis, it has two main phases. We have the interphase and we have the dividing or the mitotic phase. The dividing or the mitotic phase is further classified into two more phases, that is karyokinesis and cytokinesis. Karyokinesis is further classified into prophase, metaphase, anaphase and telophase. So we can see that in the diagram as well. So the cell will undergo interphase, followed by that it will enter into karyokinesis, first into prophase, then into metaphase, then into anaphase, then into telophase. And once the karyokinesis is completed, the cell will enter and complete the cytokinesis. So we can see this entire stages here with the diagram as well. I hope this is clear, children. Now let's move on to the next question. That is, what if the chromosomes are not aligned thoroughly at the equatorial plate in metaphase? For that, we need to understand why it needs to be thoroughly aligned. Children, when the chromosomes are thoroughly aligned at the equatorial plate during the metaphase, the division of the chromosomes is perfect. Meaning, let's say if there are 46 chromosomes at the metaphase, at the equatorial plate, 23 chromosomes will be pulled at the upper end and 23 chromosomes will be pulled at the lower end. That means there is equal distribution of chromosomes at the upper pole and the lower pole of the cell. Eventually, the daughter cells that will come from here, each will have 23 chromosomes each. So, the whole point of perfect alignment of chromosomes at the equatorial plate is to ensure that each daughter cell gets equal number of chromosomes. However, children, if this does not happen, where the chromosomes are not arranged properly at the equatorial plate, that might lead to unequal distribution of chromosomes in daughter cells, meaning some daughter cell will have more chromosome and some will have less chromosomes. Take till more chromosomes and some will have less chromosomes. And children, whenever there is unequal distribution of chromosomes in the gametes especially, it can give rise to genetic disorders. Like for example, Down syndrome. Alright children, so it is very important that the chromosomal distribution is even during cell division. Okay, I hope this is clear children. Now let's move on to the next question. A student observes the division of cytoplasm in a rose plant cell and a skin cell. In one of the cells, a structure forms in the center, whereas in the other, the cytoplasm pinches inwards. Question: name the type of cytokinesis in each cell. B. Name the phase during which the chromosomes separate from each other. And C. Write the direction of cytoplasm division in both the cells. Okay. Which are the two cells the student is studying? One is a plant cell and one is a skin cell. Alright. Now, a skin cell is obviously an animal cell. I hope this is already clear. So, now they are asking children, name the type of cytokinesis which is seen in the plant cell as well as in the animal cell. So, children, we already know about this, that in the plant cell, the cytokinesis is by a cell plate method formation. Alright, and in the animal cell, it is by cleavage burrow method. So, let me quickly show it to you as well. This is a plant cell who has a very rigid cell wall made up of cellulose. Right children? Now, since this is too rigid, it cannot bend inwards. So, in the plant cell, a cell plate is formed in the center of a cell, which will extend from center to the periphery. Alright. So, this gives answer to the last question, that is, in plant cell, the cytoplasmic division is from center to the periphery. Alright children, and in the animal cell, let's look into the diagram children. Animal cell have relatively flexible cell membrane because they do not have a cell wall, where the cell membrane will pinch inwards. It pinches inwards and separates the two daughter cells. Therefore, this movement is from periphery to the center. I hope this is clear, and this method is the cleavage burrow method. It is burrowing inside. So, we have answered the question A and C. Now, let us come to the question B. Name the phase during which the chromosome separates from each other. Children, this also we have done it a couple of times. In the prophase, children, the chromosomes are formed by condensation. In the metaphase, the chromosomes are brought to the equatorial plate by the spindle fibers. And in the anaphase, because the spindle fibers are pulling the chromosome upwards, the centromere, it breaks into two and the chromosomes are separated and they are pulled towards the pole. So, the correct answer over here is anaphase. I hope children this is clear. Yes, very simple question. Now let's move on to the next question. Match the following and the cell cycle phases. Okay, so let's quickly read the observation. Observation A, the cell prepares for division by producing protein and checking for DNA errors. Observation B, the chromosome condenses and spindle fibers begins to form. Begins is important point over here. Observation C, the cell grows, synthesis proteins and organelles. Observation D, chromosomes align in the center of the cell for segregation. And E is DNA is duplicated. So let's look at the answers. We already know these answers. Let's quickly make it. The first phase, which represents where the cell prepares for division by producing proteins and checking for DNA errors. This is the G2 phase, which is also known as the second growth phase. Coming to observation B, the chromosome condenses and spindle fiber begins to form. This is the late prophase, or simply prophase as well. Observation C, the cell grows, synthesizes proteins and the cell organelle. This is the G1 phase, which is also known as the first growth phase. Alright, coming to observation D, chromosomes align in the center of the cell for segregation. That means segregation is yet to happen. That means we are in the metaphase. And the phase where DNA is duplicated is the S phase, also known as the synthesis phase. I hope children this is clear and very simple to answer. So let's quickly look into this diagram as well. So we can see over here, this is the G1 phase, this is the S phase, this is the G2 phase. So G1 is where the cell growth happens, S is where the DNA replication happens, and G2 is where the cell is preparing for mitosis and looking for errors and checkpoints. Alright, and in the metaphase children, this is where the chromosomes will arrange at the equator before dividing. I hope this was clear children. Okay, with this, the preparation of this topic must be strengthened for you, and you might be also be feeling more confident. I am very happy to be able to make you feel more confident for your upcoming exams. Okay, now let's move on to the next topic, that is the genetics, the basic fundamentals. Now children, the expected weightage of this topic for your upcoming exam is as follows. At least 1 marker question in the form of MCQ and in the form of very short answer is expected. 2 marker question is also expected, and 1 short answer of 3 mark is also expected. That means this is a high weightage topic which is very important for us to prepare. So let's get ahead with it. Let's look at this MCQ. Pranit is diagnosed with color blindness. This disorder is an example of which type of inheritance? Option A, autosomal dominant. Option B, autosomal recessive. Option C, sex-linked recessive. And option D, sex-linked dominant. Children, color blindness is a genetic disorder. Okay, what kind of disorder is that? It is a genetic disorder. That means there is a defect in a gene. Alright, so the gene which is responsible for a colorful vision, this gene is present on the X chromosome. On which chromosome is that? That is the X chromosome. Children, X is a sex chromosome. What is X chromosome? It is a sex chromosome. So with these 2 information, you must be able to eliminate the first 2 options, right? Because these are autosomal disorders children. They are the genetic disorders that arises due to defects present in the autosomes. Now, what are autosomes? Autosomes are those chromosomes which are not X or Y. Now we know that we have a total of 46 chromosomes, right? If you are a female, that means you have 44 plus XX chromosomes. And if you are a male, that means you have 44 plus XY chromosomes. Alright. So, this 44 chromosomes, children, these are not sex chromosomes, right? Because they are neither X nor Y. These chromosomes are known as autosomes. And X or Y chromosomes, children, they are known as sex chromosomes. Therefore, option A and B are incorrect because color blindness is a disorder which is caused due to a defective gene present in the sex chromosomes. Therefore, we have two options, sex-linked recessive and sex-linked dominant disorder. Children, color blindness is a sex-linked recessive disorder. Now, let me explain this how. So, color blindness is because of a defective gene for a colorful vision present on the X chromosome. And the allele of this gene is recessive in nature. That means when we write a person has a color blindness X, we write it in this format. This means that if this is a female, she will be a carrier because this is a recessive gene. What is a recessive gene, children? Recessive genes are weak genes who cannot express themselves. They cannot express. Meaning उनकी नहीं चलती है. वो जीन नहीं काम करेगा, वो जीन एक्सप्रेस नहीं करेगा. So in this case, this female has one recessive gene, but the other gene is a healthy gene. Therefore, this weak or the recessive gene will not show itself. It will not produce any defect, and this female will be not colorblind, will be able to see properly. Okay, but in males, children, since in males we have the XY chromosomes, there is no other X. That means there is only one gene available for a good colored vision, which itself is weak and recessive. There is no other stronger gene for it. There is no other competition for it. It is just one single gene, and therefore it will express itself in males. Okay, but in females in this condition, it will not express itself because the other sex chromosome has a healthy gene for colorful vision. But in males, since they only have one X, and unfortunately this male received the recessive gene of color blindness, therefore this person will be colorblind. Okay, so with that, I hope we have got the answer that the color blindness is a sex-linked recessive disorder. I hope this is clear, children. Okay, students, now let's look at this MCQ. Which of the following results from crossing two heterozygous tall plants? Option A 100% tall, option B 75% tall and 25% dwarf, option C 50% tall and 50% dwarf, and option D all the plants will be tall. Let's solve this MCQ. So, we can see over here, children, that these are the alleles that we will receive from the parent with this gene, and these are the alleles that we will receive from this parent, right? So, let's look at the first combination. We get a homozygous tall plant. Then the second combination, a heterozygous tall plant. Third combination, again a heterozygous tall plant. And the fourth combination is homozygous dwarf plant, right? So, the phenotypic ratio, as we can see over here, there is 75% are tall plants and 25% are short plants. Therefore, the correct answer is option B. A woman who is a carrier for hemophilia marries a normal man. Which of the following statement is correct? Option A, all daughters are hemophilic. Option B, all sons will be hemophilic. Option C, 50% of the sons may be hemophilic. And option D, 50% of the daughters will be normal. Alright, children, let's look at the solution. The question asked over here is that we have a hemophilic carrier female who is marrying a normal male. Alright, and we are supposed to find out what will be the hemophilic conditions in the children. Okay, so now following the Mendel's law, we know that when the gametes are formed, only one of the allele is passed. Okay, so this will segregate into, right, and X and Y will segregate into X and Y. Now, during gametic fusions, that is during zygote formation, we may get to see the following combination. So, we may either get this combination or we may get this combination, we may get this combination or we may get this combination. So, now let us quickly write this combinations over here one more time. So, one combination is this, one is this, is XX and XY. Now, XX is a female, we know it is a recessive disorder, that means one daughter will be carrier. So, we will get a carrier daughter. Now, XY is a male, that means this will be a son. And since this is a recessive disorder, but males do not have another healthy X, they do not have another healthy gene for it, therefore, this son will be hemophilic. The other combination is healthy XX, that means this will be a normal daughter, and another son can also be, will be normal son. That means there is a 50% chance that one of the daughters will be a carrier daughter and a 50% chance that one of the sons will be hemophilic son. Yes, I hope children this is clear. Now let's look at the solution which is provided to us. So where we can see the same thing over here children. So this is a carrier female, this is a normal male, these are the gametes that will be produced, and on different combinations children, we will get one normal girl, one normal boy, one carrier girl and one hemophilic boy. Therefore, the correct answer will be children, 50% of the sons may be hemophilic. Okay. So let's quickly see why the other options are incorrect. Option A says that all daughter will be hemophilic. That was not possible. We had these two genotypes. That means we may have a carrier daughter or a normal daughter. Definitely not a hemophilic daughter. Option B says all sons will be hemophilic. Now this was also not possible. We had one normal son and we had one hemophilic son. Therefore, option B is also incorrect. Option D says that 50% of the daughters will be normal. That is also not seen. Therefore, option C is correct. Alright children, please take on the next question. Now let's look at this MCQ children. Which cross is represented in this diagram? Option A, this is the option B, option C and option D. It's a very simple question children. Let's look at the solution now. Children, we can see over here, these are the two different alleles, right, which have separated during gametogenesis. That means in one cell we had this pair of allele and in another cell before gametogenesis we had this pair of alleles, right? We know that during gametogenesis, according to the law of independent assortment, the alleles separate from each other. That means in one gamete we might just get one pair of the alleles. Alright children, therefore the cross is seen in this option, that is the option C. Alright, I hope this was clear. Now let's look at the next question. Assertion: in humans the father determines the sex of the child. Reason: the mother produces two types of gamete bearing X and Y chromosomes. Option A, both assertion and reason are true and reason is the correct explanation of assertion. Option B, both assertion and reason are true but reason is not the correct explanation of assertion. Option 3, assertion is true but reason is false. And option D, assertion is false but reason is true. let's solve this question. Children, we know that males are heterogametic in nature, meaning it has two chromosomes, that is XY, whereas females are homogametic in nature, that is it has the same sex chromosome X, right? So males are XY and females are XX. Females can only produce one type of gamete which has X chromosome in it, while the males can produce two types of gamete which can either have X or either have Y chromosomes, right? So when the sperm containing X chromosome and the ovum containing X chromosome comes together, we will get a daughter. And when sperm consists of Y chromosomes and the ovum consists obviously the same chromosome, that is X, we will get XY chromosome, that is we will get a son. So yes, since mother only produces one type of gamete, that is X, the gender of the child is determined by the father because he produces two types of gamete. Reason given over here was that the mother produces two types of gamete, X and Y, which is obviously incorrect because mother only has XX chromosomes in her cells. Therefore, the gamete can only carry X chromosomes, never Y. Therefore, children, the correct answer is option C, where the assertion is true, but the reason is false because ovum will only have X chromosomes. Okay, now let's move on to the next question. Assertion: X-linked recessive traits show crisscross inheritance, and reason: the males affected transmit the recessive allele to their daughters and not to their sons. Option A, both assertion and reason are true and reason is the correct explanation of assertion. Option B, both assertion and reason are true but reason is not the correct explanation of assertion. Option 3, assertion is true but reason is false. And option D, assertion is false but reason is true. Children, now let's look into the solution. What is a crisscross inheritance? Crisscross inheritance means this disorder, this particular genetic disorder, it moves from the father into the daughter, from the daughter into the grandson, and from the grandson again into his daughter. So it is moving from father to daughter to grandson to again granddaughter. That means the son is not affected at all. This is the crisscross inheritance. Okay, this crisscross inheritance, children, it is seen in sex-linked recessive disorders. Over here, they have taken the example of a colorblind person. Now, let me also show you the working in terms of genotype so that we understand it clearly. Here, the mother is completely healthy and the father is colorblind. All right. Now, when they get married, children, they have this particular combinations. We will have one healthy son and we will have one carrier daughter. Okay. So now we have the carrier daughter. Now this carrier daughter marries a healthy male person. Okay. In this case, what will happen, children? She will pass on this recessive gene to the son. That means the son will also have color blindness. So the father passed on the color blindness gene to the daughter and the daughter has passed on this color blindness gene to the grandson, and this grandson will further pass the color blindness gene to his daughter, who will further pass it to his son. So this is the crisscross inheritance. Okay, therefore the correct answer is that both assertion and reason are true and reason is the correct explanation of assertion. Okay. Now let's look at the next question, children. The identical pairs are collectively called as, whereas the unequal pair is known as. Now let's look at the human karyotype. These are basically the genes present in a human, right? This is the male side and this is the female side. Now we can see over here from number 1 to 22. If you look at the chromosomes, they are identical to each other. So, this 22 pairs of chromosomes which are identical, they are known as autosomes. Okay, so the first 22 children, we are talking about this first 22 for the females as well as the males. Children, but if you look at this last pair of chromosome in males, that is X and Y, you can see that they are not of the same size or structure. They are different. This unequal pair is known as the sex chromosomes. So that is why, children, you must have noticed that in textbooks or in books you must have written that males are 22 plus XY and females are 22 plus XX. Alright, XY and XX, these are sex chromosomes, whereas this first 22 pairs, they are the autosomes. Okay, children, so you can see over here the bigger diagram. This should be more clear. You can see this XYs, they are different from each other, they are non-identical. Okay, now let's move on to this next question. The shape change seen in the image occurs because of. Now let's look at the image, children. Over here we can see this is a healthy RBC which is biconcave in shape, and here we can see another RBC which is sickle shaped or half moon shaped. Now this change in the RBC shape is because of. Let's look at the gene. Children, in a healthy gene, we can see that on one strand of DNA, we have nucleotides GAG, and on its complementary strand, we have nucleotides CTC. So, when the gene is in this particular format, children, it produces a very important protein, that is the glutamic acid. And glutamic acid, alright. But when you see in this next image, children, look at this next image, the GAG gene is replaced by GTG. So, A nucleotide is replaced by T. Therefore, on the complementary strand also, instead of T, we get to see A. So, this change in its gene or in its nucleotide leads to production of a completely different protein, which in this case is valine, and because of which the RBC is a sickle shaped. This change in nucleotide occurs because of mutation. Okay, children, and therefore sickle cell RBCs gives rise to sickle cell anemia. It's a disorder that arises because of mutation where one nucleotide is replaced by another nucleotide, and as a result of which the protein which is supposed to be produced is not produced and some other protein is produced, and as a result of which the shape of the RBC changes. Okay, children, so let's quickly look at the same solution over here. So here we can see there is a replacement or there is a mutation where the A nucleotide or the adenine containing nucleotide is replaced with thymine containing nucleotide, and as a result of which the protein which is to be produced is not produced, and because of which the RBC loses its biconcave shape. Alright, children, let's move on to the next question. The position of the gene is called as dash and the differently colored segment represents dash. Children, the position of a gene is known as locus, and this different colored segment, we can see over here, this is a green and this is a purple, it represents alleles. Now let's understand that. Children, homologous pair of chromosomes, they occupy the same position on a DNA, that is, they occupy the same locus on the DNA. For that, we need to understand what is homologous chromosomes. Children, let's take this chromosome on the left side. Let's say that this particular part of chromosome, it helps in development of your eye, shape specifically. This particular part of the chromosome, it helps in development of the eye color. And this lower end of the chromosome, let's say it helps in development of your hair. Now, since we have two pairs of the same chromosomes. That means this pair, let's say we have inherited from the mother. We will also inherit a similar pair from the father, where the chromosome is present in such a way where this upper part is going to have information to develop the eye shape, this lower part is going to have the information to develop the eye color, and the lowermost part of the chromatid or the chromosome, it has information to develop the hair. This means, children, that we get information to develop the same structures from both the mother and the father. So it is never like 23 chromosomes which are coming from the sperms. They carry information to develop a body from head till the waist. And the remaining 23 coming from the mother has information to develop your body from waist till the toes. It's not the case. The 23 chromosomes coming from the father has all the information to develop your body from head till the toe, and similarly 23 chromosomes coming from the ovum has all the information to develop your body from head to toe. That means to develop one part of the body, I have two informations. Yes, and such chromosomes who have same information, who have the same genes at the same position or locus, one inherited from the mother and one inherited from the father. Such chromosomes are known as the homologous pair of chromosomes. I hope this is clear. Now coming to the second part, what are alleles? Children, alleles are variations of genes. Now let me help you understand with the same example. We can see over here this colored part. Now this is a gene for development of eye color. One is coming from mother, one is coming from father. They are present at the same locus. All that is clear. But they might not be containing the same information. That means probably the gene coming from your mother has the information to develop a blue color of the eye, and the gene coming from the father has information to develop the black color of the eye. So gene is there, it is at the same position, that is correct, but the gene is carrying a variation. It is not carrying the same information. The gene is carrying some variation, where one variation is let's develop the baby's eye into blue color, and the other variation is let's develop the baby's eye into black color. But both will help in development of the eye color. So they are genes only. Alleles are just genes, but they carry variations. They carry newer information to develop the same structure. I hope this is clear, children. Now let's move on to the next part. The 9:3:3:1 pattern, which is seen in the squares, represents the dash of a dash cross. Alright, children. So you must have solved this a couple of times. Let's quickly revise through it so that we can answer our questions. So we have taken a plant with a round and yellow colored seed, and next another plant with the wrinkled and green colored seeds. Alright, so the gametes we will have over here is this, and when we do pollination between them, we get the F1 generation with this genotype, right? Now when we are doing self pollination between them, we will get this particular set of siblings with these genotypes, right? Now we will find out the phenotypes of that. That means where there is capital R, capital Y, we know that R and Y, these are the dominant alleles, and small r, small y, these are the recessive alleles. Alright. So double R, double Y, children, this will give rise to a round and a yellow seed. This will still give rise to a round and a yellow seed. This will also give rise to a round and a yellow seed. Similarly, round and a yellow seed. Similarly, round and a yellow seed. Round and a yellow seed. Round and a yellow seed, and round and a yellow seed. So 1, 2, 3, 4, 5, 6, 7, 8, and this will also give rise to a round and a yellow seeds. That means we will have 9 offsprings who will give rise to round and yellow seeds plants. Right? Now let's look at the other combination. Here, children, we can see this will be wrinkled and yellow. Again, this will be wrinkled and yellow. Alright, and this will be wrinkled and yellow. So we have 3 progeny who will show wrinkled and yellow seeds. Next, over here children, we will get round and green seeds. Similarly, round and green seeds. So this will also be round and yellow seeds. Okay, so we will get 3 round and yellow seeds. Next, we will get wrinkled and green seed, which is only one. Wrinkled and green seeds. So what is this combination children? These are the phenotypes of the plant that we are getting to see. So the answer to the first question is 9:3:3:1. They refer to the phenotypic ratio. Alright. The second question children that was asked was, which kind of cross is this? Children, here we are considering two different characteristics of a plant, and therefore this cross is a dihybrid cross. Okay, so answer to question A was that 9:3:3:1 is a phenotypic ratio. And answer to question B is that this cross is a dihybrid cross. Alright children, now let's move on to the next question. Alright, now let's look at this question. Navneet was studying pea plants. He crossed plants with round seed and wrinkle seeds, and also tall and dwarf plants. Right, two contrasting pairs of traits Mendel studied other than the seed shape and seed color. So let's answer this first. Along with the seed shape and seed color, he also studied the height of the plant. That is, he used tall plants, which is a dominant trait, and dwarf plant, which is the recessive trait. Along with the height of the plant, he also used different colors of flowers, that is a violet flower, which is a dominant trait, and white colored flowers, which is the recessive trait. Okay. Now let's move to the next question. If a heterozygous round seeded plant is crossed with a wrinkled seeded plants, what will be the phenotypic ratio of the offsprings? So let's quickly calculate it. He crossed round seeded heterozygous plant with a wrinkled seeded plants. Right. Now here what are the possible combinations that he may get? Right. So that is he may get 50% again round seeded plants and 50% wrinkled seeded plants. Yes, that is he get a phenotypic ratio of 1:1. Right? Okay, now let's move to the next question. Which law of Mendel is demonstrated in this experiment? This is the law of segregation. Okay, now let's move ahead. Based on the given image, answer the following questions. So here we can see there is a purple flowered plant. And this is a white flowered plant. Alright, and on pollination, we get the F1 generation, which also having purple flower. Question asked over here is, name the biological term shown here. Question B, what causes this difference? Answer that. Children, the reason why there is a difference over here is variation. So the key thing that you also should be able to observe is that we have the same species of plant, but both the species are showing different colors of flowers. Here we have a violet color flower and here we have a white color flower. Alright. So what is variation? Variation, children, it is difference of traits in the same species. Let me give you an example. When you look at yourself, If you do not have any twin, do you find anybody who is identical to you? No, right? But all of them are human beings. So you all belong to the same species, but none of you look identical. And that is because of variations. Now coming to the second question, what gives rise to variations? Children, variation arises because of different alleles, right? Like for example, let's take this case. Here children, the gene for flower color is present for both the plants, but this gene has the allele capital V, and this gene has a allele small v, meaning this is a dominant allele and this is a recessive allele. So the dominant allele children will give rise to the purple color flower and the recessive allele give rise to colorless flower or the white flower. And when this flower pollinate children, we will obviously get a flower with a genotype as this. Yes or no? And since this is a dominant allele, the progeny or the F1 generation will also be violet in color. I hope this is clear children. Yes. Now let's move on to the next question. In the given karyotype diagram, identify the sex of the individuals labeled A and B. We have already solved this question ahead, children. I think you have already guessed it correctly. Yes. So yes, children, the individual with A has X and Y chromosome. Therefore, this individual will be male. And the individual with B has XS chromosome and therefore, this individual will be female. Individual with A is a male and B is the female because males have XY chromosomes, that is 22 plus XY, and female have 22 plus XX chromosomes. Right? Chalo. Now let's move on to the next question. Define genotype and the phenotype. Very simple question. Next, give an example of genotype producing a particular phenotype. Now, let's answer this question. Children, genotype is a genetic composition of an individual or an organism. And based on the information present, a phenotype will be formed. Like for example, let's say the genotype for the height of a plant is this. Here we know that this is a dominant gene and this is a recessive gene. The dominant gene always expresses itself and therefore this plant will be a tall plant. Let's do another example. If this is a genotype of a plant, then what will be the height of the plant? It will be a dwarf plant. Yes. So now with this example children, are you able to understand what a phenotype is? Yes. The phenotype is the observable characteristic of an organism. That is decided by the genotype. Now let's take a more tougher example children. This is a genotype. What will be the phenotype of the seed? Yes, you guessed it right. It will be round and yellow. Now let's take one more example. This is a genotype of another seed. What will be its phenotype? Yes, you guessed it right again. Round and green seeds. I hope this is clear children. Now let's move on to the next question. Based on the image given below, answer the following questions. Question A, identify the phenotype represented by the square labeled as follows. And next, state the phenotypic ratio of all the offsprings. So let's solve it children. Are we here? So the first question asked over here is the phenotype. So this phenotype will be dwarf plant with red flowers. Right, children? The next question is find the phenotype of all the progeny that will be seen over here. Alright. So let's look at the first one. Before that, children, please remember the T is a dominant allele for tallness, R is a dominant allele for red flower, small t is a recessive allele giving rise to a dwarf flower, and small r is the recessive allele for white colored flower. Okay, using the law of dominance, let's solve this question. So children, we can see over here, this will be a tall flower, tall and a red flower. This will also be a tall and a red flower. This will also be a tall and a red flower, and this will also be a tall and a red flower. This will also be a tall and a red flower. Alright. This will be a tall and a red flower. 6. This will be a tall and a red flower. 7. This will be a tall and a red flower. 8. And this will be a tall and a red flower. 9. So we have total 9 progenies with tall and a red flower. Alright. Now this will be tall with a white flower. This will be tall with a white flower, and this will be tall with a white flower. So total we will have 3 progenies with tall plants and white flowers. 9 progeny will be tall plants with red flowers. Alright. Now let's look at the next. We have this as the dwarf with the red flower. This is again a dwarf plant with the red flower, and this is again a dwarf plant with the red flower. So total 3 dwarf plant with red flowers. Right? And we have only one, that is dwarf plant with white flowers. Alright children, so I hope the phenotypic ratio and the different phenotypes is clear. Alright, now let's move on to the next question. Name one uncommon hereditary trait in humans and mention whether it is dominant or recessive. Next question, in Rh blood group system, which trait is dominant and which is recessive? So let's solve this question. Children, can we see this image over here? This image represents camptodactyly condition. This is a autosomal dominant disorder This is a disorder where usually the small finger, we can see this joint of the small finger, this joint remain usually flexed, meaning it is always bent. So the little finger always appear bent in such people. Okay, and this is a autosomal dominant disorder. Children, what does this mean? Dominant disorders meaning it's a dominant gene. That means it will be expressed in any individual who receives this defective gene. And what does autosomal means? That this defective gene is present in the autosomes and not sex chromosomes. Okay. The second question asked was, in Rh blood group system, which trait is positive? Children, let's quickly understand what is Rh blood group system. Rh blood group system, children, it is based on having Rh antigen or antigen D on the surface of RBC, that is the red blood cells. So if you have Rh antigen on your RBC, that means you are Rh positive. And if you do not have Rh antigen on your RBC, that means you are Rh negative. Alright children. Now Rh positive is a more commonly seen phenomenon, that is at least 80% of human population on earth is Rh positive, because Rh antigen is commonly found in the RBCs, because 80% of population have Rh producing genes in their chromosomes. Therefore, children, Rh positive is a dominant trait. I hope this is clear, children. Now let's move on to the next question. State the Mendel's law of independent assortment and give one application of Mendel's law. Let's answer the question. Children, what does Mendel's law of independent assortment says? That when more than one trait is inherited, like for example, let's say tall plants with round seeds. Okay, when more than one trait, that is tallness and round seeds, when more than one trait is inherited, the separation of the alleles, and in this case, this is a separation. The separation of the alleles and its entry into the gametes is independent of each other. We know that during meiosis, the alleles separate from each other and only one of the two alleles enter inside the gamete. When we are studying more than one traits, that is in this case the tallness of a plant and roundness of a seed. So one of the two alleles will be entering inside the gamete, but before that it needs to separate. So this separation of the alleles that takes place when we are studying two characters, it is independent of each other. That means the separation of the allele of tallness will not affect the separation of alleles of roundness of seed, and they will be independently entering into each gamete. This is the Mendel's law of independent assortment with example. Okay. Now let's move to the next question. Give one application of Mendel's law. Children, Mendel's law is so useful, that is, it helps us predict genetic disorders in the family and in the upcoming generation. Okay. So it helps us in understanding, predicting, and therefore avoiding the genetic disorders in our family. I hope this was clear. Now let's move on to the next question. Dr. Sharma examined a 10-year-old boy who suffered heavy bleeding even after minor injuries. Dr. Sharma explained the condition to the child's parents and said that the disorder is genetic. He mentioned that the males are more commonly affected. Questions: what type of genetic disorder does the boy have? Why is hemophilia more common in males than in females? And can the boy pass hemophilia to his sons? So, let's solve this question. Question A: what type of genetic disorder does the boy have? Children, here this boy is losing lots of blood just from a minor injury. That means the blood clotting is affected, and this is a bleeding disorder which is known as hemophilia. Next question is, why is hemophilia more common in males than the females? Now let's understand. Children, hemophilia is caused due to recessive gene which is present on the X chromosome, and therefore hemophilia is referred as the X-linked recessive disorder. That means that this recessive gene is present on the X chromosomes, right? Now this is a boy, that means his genotype will be this, correct? If a female also has hemophilia, for example, which is this case, because females have two copies of X. So if on one copy there is a recessive gene for hemophilia, that gene or that allele will be suppressed and the dominant healthy gene will express itself. which will form the clotting factors that will help in clotting of blood. But that is not the case in males. And the reason for that is, student, males only have one copy of X. So even if that copy has a recessive gene, since there is no countering healthy gene on Y, as a result of which this recessive gene will express itself, and therefore the male will suffer from hemophilia. Alright, so the males commonly suffer from hemophilia, but females are usually carriers. Alright, now coming to the third question. Can the boy pass hemophilia to his son? So let's quickly do this. So let's say this is a boy who has hemophilia himself and he is marrying to a female. Alright. So now let's quickly draw the combinations that we can possibly have. So we may have combination this, then we may have combination this, then we may have combinations this, and we may have combinations this. So the sons, children, as you can see, the sons are completely healthy. They are not carrying any recessive gene. Whereas if you look at the daughters, children, the daughters are going to be carriers. That means they themselves will not suffer from hemophilia, but they can pass it into the next generation. Alright, children. Now let's move on to the next question. Observe the diagram and match the labeled with their correct options. So let's look at this table. We are given this particular information. I'll give you one challenge. Let's make this diagram and calculation on our own, and then let's check it whether it's correct or not. Alright. So here we have two parents, as we can see over here. These are pure line parents, obviously. So option A, we have the parental cross with the pure lines. This is correct. Alright. Now children, when they cross with each other, the progeny that we will get, let's calculate how the progeny we will get. We will get this progeny that will be R, Y and Y. So this will be our F1 progeny. There children, we will get this progeny. So is there a question over here? Yes. Question number 3, this is going to be the F1 genotype of the progeny. Okay, now let's come to the next question. Now F1 progeny, it undergoes self-pollination on its own, right? So let's quickly make that small table. So over here, when it's undergoing pollination, we will need the male and the female gametes to see the next combinations, right? So what are the male gametes that we may get over here? So we might get this particular combination of the gamete, then we might get this combinations of the gamete, then we might get this combination of the gamete, and we might get this combination of the gamete. So yes, that is your question number 2. So these are the gametes which are produced by the F1, right? Now children, when we self-pollinate F1, we get the F2 generations, which will get us 9:3:3:1 ratio. That is option number B, which is the F2 genotypic ratio. And with that, we are just left with last option, that is the wrinkled and green seeds in F2 will be seen by the 1/16th of the offspring. Now let's look at the solution. And yes, we are correct. So let's quickly look. There is one pure line of parent who is homozygous dominant with round seeds and yellow in color, and homozygous recessive in wrinkled seeds and green in color. When they cross with each other, we get the F1 generation with this genotype. Alright. Then we got these gametes from this F1 generation, and on self-pollinating them, we will get the ratio of 9:3:3:1, where 9 will stand for round and yellow seeds, 3 will stand for round and green seeds, 3 will stand for wrinkled and yellow seeds, and 1 will stand for wrinkled and the green seeds. Right. So this will be the 1/16th of the F2 generation. Right. We are getting total 16 progeny, and only one of them will have green and wrinkled seeds. Alright children, so I hope this session was clear to you. this session boosted your confidence. It helped you understand the concepts which were weak or slightly you were confused at. And I hope you score full marks in your science paper. I will see you soon, children. Till then, stay happy and be curious.